Now let's move on to the case of euploidy or polyploidy. This is where the change of the chromosome number will involve the whole chromosome set. This is due to non-disjunction that occurs on the whole chromosome set during the process of gametogenesis. For example, let's say we have a cell here, a cell which is diploid, in which we can see that there will be two sets of chromosomes. There will be two of these long chromosomes, there will be two of these medium-sized chromosomes, and there will be two of these shorter ones. That is why we say that this cell is a diploid cell, having two sets of chromosomes. Now, when this cell undergoes the process of meiosis, maybe non-disjunction will occur on all the chromosomes. As a result, all of the chromosomes will be going into one same cell. Meanwhile, the other cell will not be getting any chromosome at all. A normal gamete should be haploid or N. However, this gamete over here is abnormal because this gamete is still in a diploid condition. And when this gamete fertilized with a normal gamete which is haploid, we will be getting a zygote which is abnormal, in which the zygote here, as you can see, will be having three sets of chromosomes, in which we can say that this cell or this zygote is showing the condition of 3N or triploid. A polyploid is a term that we use to describe a cell or an organism having 3 or more sets of chromosomes, either 3N, 4N, 5N, or maybe more than that. In this karyotype, we can see a human with 3 sets of chromosomes, in which we can see that there will be 3 copies of chromosome number 1, 3 copies of chromosome number 2, 3 copies of chromosome number 3, 3 copies of chromosome number 4, and so on, including 3 copies of the sex chromosomes. However, human triploid babies like this will normally die after being born. So the case of polyploidy is more common in the plants actually. And there are 2 types of polyploidy that we need to know. The first one will be autopolyploidy, and the second one will be allopolyploidy. Autopolyploidy shows an increase in the number of chromosome sets within the same species. Autopolyploidy occurs due to non-disjunction also, and it can be induced actually using colchicine, which is a type of mutation, a type of chemical agent that can inhibit the formation of spindle fiber during the process of cell division. Autopolyploidy is more common in the plants. For example, the banana is actually a triploid organism, 3N, or the potato, a tetraploid organism, 4N, and also the strawberries, which are actually octaploid organisms. So how can these autopolyploid organisms be produced? Let's say we have 2 individuals of the same species, and each individual will be diploid, meaning that they have 2 sets of chromosomes. Through the process of meiosis, normal gametes with the haploid condition should be produced like this, in which each gamete will be having only 1 set of chromosomes. However, when non-disjunction occurs during meiosis, abnormal gametes will be produced like this, in which we can see that this gamete over here is still in the condition of 2N. So if this abnormal gamete fertilized with a normal gamete like this, a triploid organism will be produced, in which this organism, this individual, will be having 3 sets of chromosomes. And this triploid organism is infertile or sterile. Why? Because there will not be enough pairs of homologous chromosomes for this individual to undergo the process of meiosis. Autopolyploid organisms can also be produced when non-disjunction occurs on both individuals over here. Both individuals are originally diploid, And due to the non-disjunction that occurs during the gametogenesis, abnormal gametes will be produced like this. If an abnormal gamete which is diploid fertilize with another abnormal gamete which is also diploid, then this will produce a tetraploid having 4 sets of chromosomes. This tetraploid is fertile, meaning that this individual will be able to produce gametes. Why? Because there will be enough pairs of homologous chromosomes for them to undergo the process of synapsis during meiosis. This is another case of autopolyploidy in which a diploid individual is crossed with another diploid individual of the same species, and both of these individuals produce normal gametes which will be haploid. If the normal gametes fertilize with each other, this will produce a normal diploid individual, 2N. However, with the use of colchicine, the doubling of chromosomes will occur, in which all the chromosomes will be duplicated, and as a result, a tetraploid will be produced, in which there will be 4 sets of chromosomes for this particular type of individual. So this will be a 4N individual, and this tetraploid is also fertile. Why? Because there will be enough homologous pairs for the chromosomes. The second type of polyploidy will be the allopolyploidy. This will involve hybrids produced from different species. For example, let's say we have 2 individuals, but these 2 individuals will be of different species. Let's say this individual here will be having 2 sets of chromosomes, and so this individual will be a diploid organism, and this individual will also be having 2 sets of chromosomes. This is also another diploid individual. However, their genomes are different. The genomes for this species will be different from the genome for this particular individual of another different species. And let's say these 2 individuals will produce normal gametes, in which all the gametes will be haploid. And so, if these 2 haploid gametes are fertilized with each other, a hybrid will be produced. This hybrid will be showing the condition of N plus N, in which this is a combination of 2 different types of genomes, genome set A and genome set B. This particular hybrid will be sterile or infertile. Why? Because there will be no homologous pair for all the chromosomes of A and also for B. The chromosome number in a sterile hybrid can be double due to the process of doubling of the chromosomes using colchicine, and this will produce an allopolyploid or the tetraploid, which is 4N, and this tetraploid will be fertile. Why? Because there will be enough pairs of homologous chromosomes. The allopolyploid organism is usually stronger and healthier compared to the original species. An example of allopolyploidy can be shown by these different species of grasses. We start with 2 different species of grasses. One will be the Spartina maritima, which originated from the UK, and another one will be the Spartina alterniflora, which originated from the US. This will be 2 different species with 2 different number of chromosomes, 60 chromosomes for the Spartina maritima and 62 chromosomes for the Spartina alterniflora. And these individuals will be able to produce normal gametes. The Spartina maritima will produce normal gametes with 30 chromosomes. Meanwhile, the Spartina alterniflora will produce normal gametes with 31 chromosomes. When these gametes are fertilized with each other, a hybrid will be formed, known as the Spartina tonsendae. This hybrid will be showing the combination of 2 sets of genomes. One will be the genome of the Spartina maritima and another one will be the genome of the Spartina actiniflora. The total number of chromosomes will be 61. However, this hybrid will be sterile or infertile. Why? Because there will be no homologous pair for the chromosomes. However, due to the process of doubling of chromosomes using colchicine, non-disjunction will occur and a new species will be produced known as Spartina angelica. This particular species will be a tetraploid in which there will be 4 sets of chromosomes over here, and the total number of chromosomes will be 122.